So, without going into details on this slide, there is unbelievable progress in the last 30 years, impossible to summarize in 20 minutes for patients now with CLL, NHL, and also Hodgkin lymphoma. And there are four main mechanisms, at least four main mechanisms you could think about in the broad category of immunotherapies. Targeting surface tumor antigens, that's probably the easiest thing to talk about with monoclonal antibodies. Activating tumor antigen-specific immunity, boosting immune effectors, both number and function, and then overcoming inhibitory immune suppression. And so, one can't begin to talk about immunotherapies without talking about some of the data associated with monoclonal antibodies that have been approved, particularly rituximab and anti-CD20 antibodies. There are differing mechanisms of action weighted depending on the antibody, including ADCC, direct effects, and complement-mediated lysis of cells. And probably one of the biggest, if not the biggest advance in lymphoma in the last 3 decades is the fact that our chemotherapy is a standard of care in patients with DLBCL and also follicular lymphoma. And certainly we see improvements in overall survival with the addition of rituximab in both DLBCL and also follicular lymphoma, and improved durability of responses with maintenance therapies in patients who have follicular lymphoma. And this was alluded to earlier with the mantle cell lecture, but certainly these quote-unquote chemotherapy-free next steps are potential options for patients not only with mantle cell, but also being tested in patients with follicular lymphoma in the frontline setting. So one question that's come up over and over again is whether obinutuzumab is superior to rituximab in patients with DLBCL, follicular lymphoma, CLL, and I think that I think an important thing is to not think about CLL lymphomas, although they're all lymphomas, as one group. And certainly biologically we're learning that different diseases tend to benefit from the glycoengineered CD20 antibodies than others. And in CLL, I think, if you think about the CLL-11 study, there's certainly a clear benefit to the obina over the rituximab based on that particular design. In DLBCL, we saw the Goya study presented, which was the OCHOP versus RCHOP, with the primary endpoint being progression-free survival. And here the glycoengineered antibody didn't add a whole lot in terms of progression-free or overall survival to the chemotherapy backbone. And so at least in DLBCL, RCHOP remains the standard of care for all patients. In follicular lymphoma, there's a little bit of a different answer that came from ASH this year. These are the 5-year survival data for these so-called early progressors with follicular, and there are certainly groups of patients with follicular lymphoma who are an unmet clinical need with early progression and inferior survival after standard immunotherapy or immunochemotherapy combinations. We saw the Gallium study presented, which was obina versus Rituxan with chemo backbones, including CHOP, CVP, bendamustine, with a maintenance required for all patients with obinutuzumab versus rituximab. And although there weren't tremendous differences in the overall response rates or the complete response rates, what we did see was a difference in the durability of response with the obina versus rituximab with chemotherapy backbone. And here it's listed at the presentation of 24 versus 16.8 months, and you can see there's a difference in 3-year progression-free survival. There may have been some subtle differences in terms of the toxicities of the chemotherapy backbones, with the bendamustine arm seeming to lead to more toxicity and deaths than the other backbones, but that wasn't the point of the study. The point was to ask whether or not PFS was extended by a different CD20 glycoengineered antibody, and that primary endpoint was met. There are many targets available in Hodgkin and non-Hodgkin lymphoma. I'll mention anti-CD30 drug conjugate, brentuximab, which I'm sure everyone in the room has used in both patients with Hodgkin and anaplastic large T-cell lymphoma. It's a CD30 targeted antibody conjugated to MMAE, and we saw in 2012 originally and 2010, when these data were presented in patients with PTCL and Hodgkin lymphoma, an incredibly active immunoconjugate for both of those patient populations. There are long-term data that were presented at 2 ASHs ago and this ASH for patients with Hodgkin lymphoma who received brentuximab, and in this 3-year follow-up, of the 34 patients who achieved a CR, 47% were still without evidence of disease at 53 months. And then to extend that a little bit further into the future, even with 5-year follow-up data, there is a small subset of patients who have an incredibly durable response in the setting of relapsed refractory Hodgkin lymphoma, with the curve appearing to flatten and one could imagine that although we can't identify who these patients are at the subset, that there may be opportunity for even long-term remission and or cure with immunoconjugates in Hodgkin lymphoma. I'll now mention activating tumor antigen-specific immunity and just mention the fact that there have largely in the pre-rituximab era been vaccines developed. Steve Schuster from Penn presented data in 2013 at the ASCO meeting for the BioVest vaccine in follicular lymphoma, and certainly an interesting area, but not the major discussion of today's lecture. I'll next move to the topic of boosting immune effectors, number and function of cell therapy, and certainly the model that one thinks about is harnessing the immune system to treat lymphoma is allogeneic stem cell transplantation with all its limitations. And over the last couple of years, first in CLL and then in ALL and then in NHL, we've seen autologous and also allogeneic chimeric antigen receptor T cells that are specific against CD19 being employed and quite active. I won't talk in tremendous detail about CAR T cells because there'll be a subsequent lecture, but I'll mention some of the data that have come out of the group at the University of Pennsylvania under Steve Schuster. This is the rationale for CD19 as a target. This is a schematic that I believe was designed by David Porter and Carl June, looking at what happens in terms of lentiviral infection to T cell expression of the chimeric antigen receptor that's specific to CD19 with a co-stimulatory molecule for 4-1BB, recognition of the target, and then apoptosis of the target. There are different chimeric antigen receptors termed first, second, and generation. All of the data that have been presented so far over the last couple of years have been from second-generation CARs that have one co-stimulatory domain. Third-generation molecules have two co-stimulatory domain. And the first paper that came out of Penn was authored by David Porter for 3 CLL patients, and this had tremendous impact in terms of the popular press and gaining attention for this potential treatment modality. Steve has led the charge at Penn in terms of treating over 50 patients with NHL with CAR T cells and has, I think, one of the longest follow-up series of patients. This was the design of the study, patients with relapsed refractory disease. And I won't go into all the details except to say that this included patients with DLBCL, follicular lymphoma, and mantle cell lymphoma. This was the patient allocation, 43 patients. The 26 patients had DLBCL that have been previously presented, heavily pretreated sick patient population, and I think all of these CAR studies probably underestimate how sick these patients are at the time that they're treated. But the data that Steve presented at 3 months was an overall response rate of 47%, and with long-term follow-up, best response of 47%, with some PRs converting to CRs over time. The swimmer's plot that he presented demonstrates how durable some of these responses can be. And then this is the progression-free survival of 3 months overall for the series, but for those responders, and again, predicting who those people will be are very important. And Steve is doing a lot of work in collaboration with scientists at Penn to identify those patients who will respond to CAR T. You can see quite durable responses with a median follow-up here of 14.5 months, but it's much longer at this point in time. In follicular, a similar story, heavily pretreated patient population, median prior therapies of 5, and the overall response rate in folliculars was much higher, 73%. One could argue maybe that's because of the pre-CAR chemotherapy concoctions, but essentially these patients were all chemo-refractory patients, and with a long-term follow-up, you wouldn't expect to see data this good with a single round of chemotherapy. We wish it could be the case. The responses are also durable on the swimmer's plot, and you can see here with 11.9 months follow-up that the median progression-free survival was not reached. I won't talk about it, but I suspect it'll be discussed today that the KITE phase 2 Zuma trial was presented at ASH this year, which also had very interesting results. I'll next move to the realm of checkpoint inhibitors and focus on patients with NHL and Hodgkin lymphoma. I already mentioned that there's probably a role for checkpoint inhibitors in Richter's and probably in CLL as well, although not as monotherapies. Programmed cell death inhibition in lymphoma is recognized as a very important pathway for inhibition. There are 2 agents that have gained the most attention, PD-1 inhibitors, pembrolizumab and nivolumab, which is approved in relapsed Hodgkin lymphoma. There are others that are being studied. And certainly T-cell inhibition by PD-L1 and PD-L2 expressed by lymphoma cells is an important way of deactivating the immune system, and anti-PD-1 antibodies and anti-PD-L1 antibodies, for that matter, have the ability to block that signal and reactivate the immune system. There are data presented as far back as ASH 2014, which are compelling in patients with both Hodgkin lymphoma and non-Hodgkin lymphoma, where we can even see across the board as monotherapies that these drugs can be quite active for patients. The most compelling data is in Hodgkin double refractory patients presented in New England Journal of Medicine of nivolumab. Overall response rate initially reported at 78%. 17% of patients had CR. These are Hodgkin's therapies who are beyond Hodgkin patients who are beyond brentuximab and beyond autotransplant. There are other PD-1 inhibitors that have been presented for patients with DLBCL. This is a grouping of patients who were treated after autotransplant. And the most compelling figure here is that PET positive survival curve for patients, where those patients post-autotransplant, especially in an era where everyone's been treated with rituximab, are expected to have a dismal prognosis based on data that one might see from the CORAL series. But here we're seeing a response rate that appears to be durable at 51%. And those are patients who have measurable disease after autotransplant, and the drug is also well tolerated. So an interesting concept for high-risk patients following autotransplant with DLBCL. Activity has also been reported in follicular lymphoma, where we're seeing high response rates that appear to be durable. And at ASH, there were some updated data presented for checkpoint inhibitors in Hodgkin lymphoma. This is the phase 1 trial of pembrolizumab in Hodgkin, the KEYNOTE-013 study. And again, we're seeing response rates that are quite high, 58% in high-risk patients with a CR rate of 19%. When we look at the phase 2 data, the KEYNOTE-87, you can see, if I can find my pointer, across the board, patients who were progressed after auto and BV, patients who failed salvage therapy, ineligible for auto, and then patients who failed auto and not treated with BV after transplantation, that response rates are in the 50 to 70, almost 80% range, a high percentage of patients with CRs, and when you look at these patients, the responses appear to be quite durable. So a very active mechanism of inhibition in patients with Hodgkin lymphoma. This is longer-term follow-up of the phase 2 data of nivolumab, the CheckMate 205 study, and here we see progression-free survival curves that look quite interesting for patients, especially patients who have a PR can also have quite durable responses to these medications. We're starting to look at combination therapies, and these are Hodgkin patients who receive brentuximab plus nivolumab. Very compelling, although early data, where we're seeing response rates of 90% and CR rates of 62%. And just to conclude about this aspect of immunotherapies, there are numerous PD-1 inhibitors in trials. We're now starting to see PD-L1 inhibitors as well, and there are other immune checkpoint inhibitors, for example, anti-LAG inhibitors that are being developed. Combination therapies are being tested in NHL and also Hodgkin lymphoma, and studies that are post-transplant are ongoing and potentially quite compelling. Steve, as always a forerunner, has identified patients who failed CAR T cells early on and is now combining pembrolizumab, or administering pembrolizumab in those patients with very interesting correlative data to soon be presented or has recently been presented in blood, and a clinical trial that's ongoing at this time. Finally, I'll mention bispecific antibodies, which I think are probably one of the most interesting areas in lymphoma and CLL and are probably underdeveloped at this point in time. These have the combined specificity for a target and endogenous effector cells. They were developed as early as 1990, so this isn't exactly a new concept, but an antibody that bridges the immune effector to the target is certainly very appealing, not only because of potential activity, but because these are off-the-shelf products that could potentially be used in all patients. This is just a cartoon of a bispecific T-cell engaging antibody, a BiTE, bridging two T cells to a CD19 positive tumor cell. This could be ALL, CLL, DLBCL. There are preclinical data dating back as far as 2000 for the development of these particular antibodies, where there is incredible rapid high lymphoma-directed cytotoxicity in unstimulated T cells. So the preclinical data has been around for nearly 2 decades. Blinatumomab was developed and is primarily approved at this point in time for patients with ALL. It links CD3 polyclonal T cells to CD19 expressing B cells. It's constructed with 2 scFvs, bypasses TCR specificity and MHC1. And the appealing aspect of this molecule is that it induces T cell activation, T cell mediated B cell lysis, potentially has the effect of of memory T cell expansion, and serial target cell killing, so you don't necessarily, unlike a CAR T cell strategy where most patients receive a CAR once, you can give these over and over again to induce a response. There are preclinical data dating back as far as, clinical data dating back as far as 2008, where we saw the first confirmed response to single-agent blinatumomab in human subjects, and there are phase 2 data that have been presented now for patients with ALL, where in these refractory patients where the chance of getting to transplant and or surviving for a year beyond the diagnosis was less than 10%, we're seeing 80% MRD with median follow-ups of 33 months and CR rates of 69% with long-term follow-up. So quite impressive in ALL, which led to the approval. There were a couple of papers that were published last year looking at blinatumomab in patients with DLBCL. This was a multi-center phase 2 trial published in 2016, 3x3 design, all subtypes, but really we focused the most on the DLBCL patients because they have the largest need with the dose expansion at the MTD. The results here have been quite impressive. They appear to be durable, and if you look at patients with DLBCL, the targeted dose is 60 micrograms per meter squared per day. A window into the toxicity here is similar to what one sees in CAR T cells. Neurotoxicity is the most common AE, 71% overall, 22% grade 2 or higher. Fortunately, they occur relatively early. There are strategies being developed with corticosteroids to mitigate this risk, and all neurological events in this particular series were self-limited, albeit some lasted up to 29 days, and later events were not reported. This is a window into the activity of blinatumomab in this series. You can see that the overall response rate was 69%. In the DLBCL subset, it was 50% of patients. Some responses can be quite durable. Median response duration was 404 days for this patient population. And this is just, one of those examples where a picture is worth 1,000 words. For blinatumomab monotherapy, you can see imaging at baseline with a large measured node, follow-up, and end of study. There's a second series published in 2016 looking at 2 sequential strategies. One was a stepwise dosing strategy, one was a constant dosing strategy. The overall response rate reported here was 43% in patients with DLBCL. There's also interesting data presented in the paper with the use of dexamethasone to mitigate neurological toxicities. You can see here that there's significant reduction in lymphadenopathy. And in this series, for responders, the median duration was not reached, although overall, the median overall survival for the patient population was 5 months. I will mention that blinatumomab is not the only bispecific, and there are literally hundreds of bispecific antibodies that are in development. There's also the concept of a DART, which has been implemented in several clinical trials, which is a dual affinity retargeting molecule. It's a little bit different in its chemical structure than a bispecific antibody, but whereas blinatumomab is a continuously infused molecule, which is cumbersome and difficult to give to patients even with ALL, DART antibodies and some other concepts are rapid infusions over 30 minutes, which can be given every 2 to 3 weeks. So that's potentially an advantage if we're seeing similar activity. So in summary, and hopefully I stayed on time, for immunotherapies, immunotherapy is the 4th modality in cancer treatment, many promising and likely game-changing applications in lymphomas. I think immunotherapies are certainly, in the largest part, the future of where therapy is going. Will help to offer non-chemotherapy options, particularly to patients with indolent disease, and in high-risk and aggressive diseases like DLBCL, for example, they provide a platform consolidation and maintenance post-induction. I'll stop there.